Okay, let's get into this. It sounds almost unbelievable, doesn't it? Finding a human brain, thousands of years old, but still preserved.
It really does, like something out of fiction. But yeah, under certain very specific conditions, soft tissues like brains actually can survive for ages.
And we're talking about real examples here, like from that excavation site in Bristol, UK, Blackberry Hill Hospital.
That's the one, an old hospital, also a prison site, I believe, and they found potentially hundreds of these ancient brains there. It's just an incredible, maybe slightly unsettling find.
Definitely unsettling. And getting useful biological info out of ancient soft tissue, well, that's always been the really hard part, hasn't it? We hear a lot about ancient DNA, usually from bone or teeth.
Right.
So this deep dive, it's focused on something else. Ancient proteins, paleoproteomics.
Exactly. And proteins are, well, they're crucial. People say DNA give you the potential, the blueprint.
But proteins give you the record, what was actually going on in the body.
Precisely. What was being built, what functions were happening. And the key thing for ancient remains is proteins can often last way longer than DNA does, especially in certain conditions.
But the challenge, specifically with soft tissues, like the brain, it's just packed with so much more protein variety compared to bone, right?
Massively more. The brain has something like, 76% of all human proteins. Bone has less than 9%. So there's this enormous library of biological information in preserved organs that we've struggled to really tap into.
Because getting those proteins out is tricky.
Extremely tricky. Bone proteins are one thing, but soft tissues, they're complex mixtures. You've got all these different types, especially those tough membrane proteins, all tangle up in degraded ancient material. It's a real extraction nightmare.
And that's the core problem this research paper we're looking at decided to tackle head on. They wanted to find a much, much better way to get proteins out of ancient soft tissues.
Yeah, using these archaeological human brains as the perfect, challenging test case.
So our mission today for you listening is to unpack how they did it. What's this new method? What did they learn about how proteins even survive for millennia? And crucially, what can these ancient brains now start to tell us?
Let's dive in.
Okay, so the starting point. This specific sample from that Bristol site. A small piece, a biopsy, from the outer layer of the cortex of one particular brain. Identifier SK1T999, and it was apparently quite well preserved.
Relatively speaking, yes. But even well preserved for ancient soft tissue means it's incredibly fragile and degraded compared to fresh tissue. And the first big hurdle, the real bottleneck, is just getting the proteins out. Sample prep.
Right. Like you said, it's tough, especially for those membrane proteins that are kind of greasy and embedded.
Exactly. They're all mixed up with fats and other molecules, and in ancient tissue, they often clump together, aggregate. You need strong chemicals to break all that apart.
Chemicals like urea or STS detergents, basically.
Yes. But the catch-22 is that those very chemicals, the ones good at breaking things open, can then interfere with the next stage, the actual analysis, where you identify the proteins. They can gum up the works, so to speak.
Ah, okay. So the researchers had to find a balance. How did they approach that?
Methodically. They didn't just guess. They tested 10 distinct protocols, 10 different ways of extracting and cleaning up the proteins from this ancient brain sample.
10. Wow, so comparing different approaches.
Exactly. They tried different initial extraction buffers, chemicals like urea, STS, sodium lauryate, which are designed to break open the cells and dissolve proteins.
Okay.
And then they combined those with different cleanup methods, ways to get rid of the salts, the detergents, anything that wasn't protein before analysis, things like SPE, FASP, S-trap, SP3, different techniques for isolating the target molecules.
So like testing different combinations of dissolve and purify steps to find the magic recipe.
You got it. A systematic search for the best possible workflow for this unique, challenging material.
And once they had potentially extracted these proteins, they needed some serious tech to figure out what was actually there, right? LC FAMES, MSMS. Sounds complicated.
It is powerful stuff. So LC-MSMS is pretty standard in proteomics. Liquid chromatography separates the protein fragments, and then mass spectrometry identifies them by measuring their mass.
Okay, got that part. What about the FAMES bit?
Yeah.
High field asymmetric waveform ion mobility spectrometry. That's the extra ingredient here.
That's the key addition. FAMES adds another layer of separation before the protein fragments even get to the mass spectrometer.
How did that help? Another filter.
Exactly. Think of it as a pre-filter or an extra sorting gate. It uses electric fields to separate the ions based not just on mass and charge, but also on their shape and how they move through a gas under that field.
Okay.
And what that does, especially for ancient samples, which are, let's face it, chemically messy,
Right, full of junk from decay and the environment.
it filters out a huge amount of that chemical noise, those interfering molecules that aren't actually peptides. This makes it way easier for the mass spec to see the real protein signals, especially the ones that are present in only tiny amounts.
So it boosts the signal by cutting down the noise.
Massively. They found that adding FAMEs increased the number of unique proteins they could identify by up to 40%. That's a huge jump in sensitivity.
40%, just from adding that extra filtering step. Okay, so after testing all 10 methods, which one came out on top? What was the winning formula?
The clear winners were the protocols that started with urea-based extraction.
Urea. Why urea?
Urea is really good at, well, denaturing proteins and breaking apart membranes and those stubborn protein clumps, those aggregates I mentioned. That's vital for getting inside the cells in dense, aged tissues like the brain.
So it really opens everything up.
It does. And because of that, they could access proteins from deep within the cellular machinery. They specifically mention finding parts of the mitochondrial ATP synthase complex, that's the energy-producing engine of the cell, which were much more abundant using the urea methods.
So urea gets you access, and then FAMES cleans up the signal.
Exactly. That combination, ureolysis, followed by LC-FAMES MSMS analysis, that was the powerhouse. It allowed them to identify, quote, the largest and most diverse collection of ancient proteins ever recovered from archaeological material.
The largest ever. How many are we talking?
With the best protocols using urea combined with either a cleanup method called FASP or a commercial kit called the Preomics Beatbox, they identified over 1,000 unique proteins.
1,000 from one tiny ancient brain sample.
Yeah, and compare that to some of the other methods they tested. Like one using a technique called SP3 only yielded about 104 proteins. So it's literally an order of magnitude difference. From just over 100 to over 1,000. That's the scale of the breakthrough here.
That's incredible. Just unlocking so much more information. But okay, identifying what proteins are there is one thing. What did the state of those proteins and which ones survived best tell them about the whole process of preservation and decay over thousands of years?
Ah, yes. This is where it gets really interesting biologically. They found some clear biases. First, a preservation bias. It looked like hydrophobic proteins were sticking around preferentially in the ancient brain.
Hydrophobic meaning water avoiding, like membrane proteins.
Exactly. Proteins often found embedded in cell membranes or forming structural components. They found that about 40% of the protein fragments, the peptides they recovered, were hydrophobic. Compare that to a modern living brain proteome where it's only about 13%.
Wow, quite a difference. Why would the water-hating ones survive better?
Well, one strong hypothesis relates to the decay environment itself. As tissue breaks down, microbes and chemical reactions produce acidic byproducts. The pH drops. In that more acidic environment, hydrophobic proteins tend to clump together, to aggregate, and become less soluble in water. This clumping might actually shield them, protect them physically and chemically from further breakdown over long periods.
So the decay process itself might inadvertently help preserve certain types of proteins. Makes sense.
But then there was another twist, a retrieval bias. Even though the hydrophobic proteins seemed better preserved overall,
Yes.
the methods they used, especially the successful urea-based ones, tended to be better at actually extracting and identifying peptides that were more acidic, peptides with a lower isoelectric point, or PI.
Huh. So water-hating proteins survive better, but acidic proteins are easier to get out with the method. How does that work?
It seems counterintuitive at first, but it likely ties back to the same decay chemistry and the nature of the surviving proteins. That acidic environment created during decay might help preserve some proteins, but it also influences which ones are most easily solubilized and extracted by the lab chemicals later.
And did they see particular types of proteins fitting that acidic profile?
They did. They found a significant enrichment of cytoskeletal proteins. Think of the internal scaffolding of the cell, things like tubulins and intermediate filaments. These structural proteins are known to be generally more acidic and also less soluble to begin with. So their inherent properties might make them both relatively resistant to decay and more readily retrieved by these extraction methods, especially compared to other protein types. It might explain both the preservation and retrieval patterns they observed.
So those structural proteins are kind of double winners in this scenario. They survive well and they're easier to detect.
It seems that way. They appear to be really robust markers in these ancient brain samples. And beyond just which proteins were there, the researchers also looked for molecular scars, signs of the immense time that had passed.
Like chemical changes to the proteins themselves.
Exactly. What scientists call diagenetic modifications, changes happening after death. A common one is deamidation, where an amide group is lost from certain amino acids.
And did they see that?
Oh, yes. They saw specific patterns. For instance, deamidation on the amino acid arginine seemed particularly linked to proteins from the myelin sheath, that fatty insulation around nerve fibers.
Interesting detail. Any other signs of aging?
They also found quite a few non-tryptic peptides.
Non-tryptic, meaning they weren't cut where the enzyme trypsin usually cuts in the lab.
Precisely. Trypsin is the enzyme normally used to chop up proteins into identifiable fragments in proteomics. Finding fragments with ends that don't match where trypsin cuts suggests the protein chain broke apart spontaneously over time due to decay processes.
Ah, natural degradation breaks.
Exactly. And they noted that myelin proteolipid protein, another component of that nerve insulation, was a frequent source of these non-tryptic fragments. It gives clues about which proteins are maybe more susceptible to just falling apart over millennia.
Makes sense. Now, you mentioned messy samples earlier. What about contamination? Always a risk with ancient stuff, right?
Always. You have to be vigilant. They did check thoroughly for common lab contaminants. PEG polyethylene glycol, a common polymer, popped up as the most abundant potential contaminant in pretty much all samples.
Okay.
And they also noted that the commercial kits, while convenient, sometimes seem to introduce a slightly broader range of contaminants, like various surfactants, compared to the methods using purely lab-made solutions. It's just part of the process. You identify contaminants and filter them out of your final analysis.
Good to know they accounted for that. Now, there was one limitation they mentioned, right?
Yeah.
About the number of samples.
Yes, and it's an important one. Because archaeological human remains are, quite rightly, precious and irreplaceable, they performed this extensive comparison of 10 methods using subsamples, all drawn from that one brain biopsy, SK10999.
So it's an incredibly detailed look at that specific sample, but not necessarily representative of all ancient brains everywhere.
Exactly. It was essential to do it this way to get a reliable comparison between the 10 methods you need to use the same starting material. But it means this study is primarily a proof of concept. It shows the method can work incredibly well on this type of material.
But more work is needed on other samples.
Definitely. Future studies will need to apply this optimized workflow, the urea extraction plus FAMES, to brains from different individuals, different archaeological sites, different time periods, to see if the specific patterns of preservation and decay they found here are universal or if there's variation.
Still, the fact they got over 1,000 proteins from, less than 50 milligrams of tissue?
Yeah.
And they only injected a tiny fraction of that.
Yeah, they mentioned only injecting about 5% of the processed sample. That's really promising. It shows you don't need huge amounts of this precious material to potentially get incredibly ripple data using this approach.
Okay, let's pull it all together then. What's the big takeaway for you, for the listener? Why does optimizing protein extraction from ancient brains really matter?
It matters because it fundamentally changes what we can learn about the past. This optimized method is like finding a key to a locked library. A library full of detailed biological information stored in soft tissues, especially the brain, which is just so incredibly rich in functional proteins.
Information we just couldn't reliably access before.
Right. We're moving beyond what bone alone can tell us. Bone is great for DNA, for certain structural proteins, but the brain, other organs, they reflect so much more about the individual's actual lived experience, their physiology.
So we could potentially learn about health, diet, maybe even diseases.
Absolutely. Imagine being able to look at the proteins in a preserved ancient heart or liver or kidney. You could get unprecedented insights into metabolic conditions, infectious diseases, environmental exposures, nutritional status, things that were previously invisible in the archaeological record. It shifts our focus from just identity and ancestry towards understanding the actual biology of people who lived long ago.
So to sum up this deep dive, the big achievement here is this new, refined toolkit, basically using urea to break things open effectively, and FAMES to clean up the signal, which lets us finally tap into the huge store of protein information in ancient soft tissues.
Yep. And in developing that tool, they also gave us some fundamental insights into how these proteins actually survive for millennia. The biases involved hydrophobic ones sticking around, acidic ones being easier to retrieve, and the chemical fingerprints left by time and decay.
It really opens things up now that we can access this level of detail from ancient brains, ancient organs. It makes you wonder, doesn't it? What questions about human history, about ancient health crises or adaptations, questions we couldn't even properly ask before, might we now be able to tackle?
Exactly. What other secrets are hidden in these preserved tissues, just waiting for the right methods to bring them to light? The potential is really quite staggering.